तस्य रूपमधिकम् प्रच्छन्नगुप्तं धनम् विद्या भोगकरी यशः सुखकरी विद्या गुरुणा गुरुः विद्या बन्धुजनो विदेशगमने विद्या परं दैवतम् विद्या राजसु पूज्यते न तु धनम् विद्या विहीनः पशुः विद्या प्रशस्यते लोकैः विद्या सर्वत्र गौरवा विद्यया लभते सर्वम् विद्वान् सर्वत्र पूज्यते ज्ञानेन सदृशं पवित्रमिह विद्यते न हि ज्ञानेन सदृशं पवित्रमिह विद्यते न हि ज्ञानेन सदृशं पवित्रमिह विद्यते।
Dr. Amita Kocharekar is a dedicated and accomplished assistant professor in the Department of Microbiology at Sathaye College, Mumbai. With over 4.5 years of experience in academia, she has made significant contributions to the field of microbiology, particularly in the areas of microfluidics and the development of low-cost point-of-care devices. With a doctoral degree in microbiology, Dr. Kocharekar has delved deep into the areas of research methodology and scientific inquiry. Her doctoral thesis is focused on pioneering research in the synthesis of flavoring compounds employing lipase and microfluidic systems. She has cleared SET and NET exams too. Throughout her academic journey, Dr. Kocharekar has demonstrated exceptional academic excellence, earning distinctions in both her master's and bachelor's degree in microbiology. Her research endeavors have been published in esteemed scientific journals, highlighting her commitment to advancing knowledge in her field. Dr. Kocharekar's research interests span a wide range of areas within microbiology, including enzyme optimization, microbial detection methods, and bioprocess engineering. She has actively participated in national and international conferences, contributing to the dissemination of scientific knowledge and fostering collaborations within the scientific community. In addition to her research pursuits, Dr. Kocharekar is deeply passionate about teaching and mentoring students. As an assistant professor, she has demonstrated excellence in classroom instruction, curriculum development, and student engagement. Her commitment to fostering a stimulating learning environment has earned her admiration and respect of her students and colleagues. Outside of academia, Dr. Kocharekar is actively involved in extracurricular activities, including research presentations, workshops, and industry visits, further enriching her academic experience and professional network. With her expertise, dedication, and passion for microbiology, Dr. Amita Kocharekar continues to inspire and empower the next generation of scientists at Sathaye College, shaping the future of microbiological research and education in India. Let's welcome Vidyavachaspati Dr. Amita Kocharekar.
Namaste. I am Dr. Amita Kocharekar from Department of Microbiology of Sathaye College. I extend my heartfelt gratitude to our esteemed management at Parle-Tilak Vidyalaya Association for providing me with the opportunity and platform to share my remarkable journey of my doctoral research through the Vidyavachaspati program. It is with a great honor that I am here today to recount the tale of my research journey, which was marked by profound discovery, formidable challenges, and ultimately triumphant achievement. The main motto to choose a topic of the doctoral research was that there is lot of wastage of reagent, samples, and there is a need for rapid detection technology. There are people living in some rural area where they cannot afford quick laboratory facilities and they need to rush to laboratories for diagnosis. So my motto was to make some biosensors related microfluidics devices which can estimate the analyte on site. My journey commenced at Guru Nanak Khalsa College under the mentorship of Dr. Meenal Dukhande, Assistant Professor from Department of Microbiology. With her guidance, I embarked on a quest to explore the fascinating field of flavor synthesis using lipase enzyme. The title of my doctoral research was A Study on Synthesis of Flavoring Compound Using Lipase and Microfluidic System. Now, there are lots of flavoring compound available in the market, but all these flavoring compound are made using chemicals. And since flavoring compound are used all where there is a huge demand of flavoring compound. In an era where the demand for processed and preserved food is on the rise, there is also a parallel demand for enhancing their palatability. So not only the customer is interested in the smell, but also they want the palatability. And as the consumer has become increasingly aware of the importance of natural flavor, the quest for alternative strategies in flavoring production has also increased. So my main goal was to make use of some biosynthetically synthesized flavoring compound which is not going to be harmful for humankind. Traditionally, the flavoring compound which are used, they are either extracted from natural material like plant extract or they are extracted from animal. But as we know that it is not a sustainable solution, I tried to use lipases, a biocatalyst, to give a biotechnological alternative and best solution for synthesizing this flavoring compound. There are huge amount of enzymes available on the earth, but I tried to utilize lipases. Lipases are the unsung heroes of the flavor synthesis. Lipases catalyze lot of reaction, but generally they are known for transformation of lipid into aromatic compound by esterification and trans-esterification reaction. With this reaction, they contribute to the huge amount of bouquet of the flavor which we encounter. And there was very less amount of work done on lipase. Now lipases are produced by microorganisms, they're also produced by animals and all, but since we wanted some sustainable solution, I tried to use microbial source for lipase production. The objectives of my study were multifaceted, aiming to unravel the secret of lipolytic organism, optimize condition for enhancing lipase production, further purify lipase and explore their immobilization, and ultimately I have used this enzyme in esterification and trans-esterification reaction to produce flavoring compound. Not only this, but I also tried to use microfluidic technology for rapid detection of lipase activity and also for rapid detection of esters. As we know that flavoring compounds are aromatic compound, there are huge categories in this compound. The flavoring compound can be alcohol, it can be aldehyde, it can be ketone, acid, esters, phenol, and etc. However, among all this, esters are mainly known for giving that distinct aroma for the compound. Esters are generally fruity to smell. They have a sweet fruit-like odor, and they are made up of short chain of esters. They are made by condensation reaction between alcohol and carboxylic acid. Now, lipases have tremendous application. They are used in food industries for development of flavors in milk, cheese and butter. They are also used in bakery industries for flavor improvement. They are used in beverages for improving aroma of the drinks and all. They are also utilized in detergent industry for cleansing action, for removing stains and dyes. This is not the only thing which our lipase are used. They're also used nowadays in biodiesel production. Yes, they're also utilized in fuel production. Further, they are also utilized in pharmaceutical companies for hydrolysis of alcohol. They also have application in cosmetic brands since they are having a sweet fruity odor. They are used in nail paint remover, in manicuring technique, and they are also nowadays used in meat and fish technology for flavor development. Whenever these meat and fish articles are exported, there is a need to maintain flavor. So in this technology, lipases are used. So we can understand that lipases has use application. Now, to begin with my journey, the initial phase of my study involved isolating lipase producing bacteria from diverse environmental samples. Okay, I had used spoiled milk sample, spoiled milk, meat sample, spoiled fish sample, dairy samples and all, oils and grease sample. So I have scanned many environmental sample for taking lipase as an lipase to screen for lipase producing enzyme. Through meticulously screening, I further went for taxonomical identification. So the organisms which were giving a good amount of lipolytic activity were further identified using bioinformatic platform which involve FASTA and BLAST technology. Now, this process requires not only technical expertise but also a keen understanding of microbial ecology and taxonomy. Further, in order to ensure lipase activity, different lipase detection methods were standardized. So, detection of lipase activity was mainly carried by colorimetric assay, which include para-nitrophenol dodecanate method and 4-amino antipyrine method, and I also went for titrimetric assay which include olive oil emulsion method. So the main principle of para-nitrophenol dodecanate method was it involved the use of chromogenic substrate which is para-nitrophenol dodecanate. Now lipase enzyme cleaves this substrate and thereby releases para-nitrophenol which gives yellow coloration. So as the concentration of yellow coloration increases, which can be detected by colorimetrically, we can quantitate the amount of lipase activity. Apart from para-nitrophenol dodecanate method, we have also used 4-amino antipyrine method. Herein, phenol reacts with 4-amino antipyrine in an alkaline solution to form a dye complex in the presence of potassium ferrocyanide. And the titrimetric method is like acid-base titration, wherein fatty acid released from olive oil due to the action of lipase is determined using NaOH and phenolphthalein as an indicator. So the potential lipase producers were identified using bioinformatic platform, and the sequences which were obtained, they were novel sequences. Yes, out of around 14 to 16 isolate, there were 3 isolate, namely Staphylococcus hominis, Bacillus altitudinus and Staphylococcus galerium, which were novel and which was firstly isolated from our team. So the sequence of this isolate was sent ahead for gene bank at National Center for Biotechnological Information, and after that we get the accession number. So this bacterial sequences, that ATCG sequences, that DNA sequences will be all time deposited in the library. So next time if anyone is sequencing the same entity, the person is going to get the sequence pathogen and we are going to be the author of the same. So this is a major outcome of my study, wherein we can see that the potential sources were identified. Further, after standardizing the lipase detection method, we went ahead with optimization of lipase production. Now, optimization include changing formulation of the growth medium. And not only growth medium, we have to standardize the amount of organism added for production of the enzyme. So whatever the three potential isolates were there, I changed their optical density from 0.1 to 0.25 OD. And I then monitored the effect of changing optical density on the production of lipase and thereby lipase activity. Not only optical density, further pH was also standardized. pH was standardized from 5 to 9. And not only this, we went on step by step standardizing different parameter and thereby then compiling all parameter 1 and then to see whether lipase production has enhanced or not. So after pH, we varied temperature from 25 degrees centigrade to 45 degrees centigrade. And there was observation that lipase production require agitation. Okay, it required oxygen since the bacteria are aerobic. So we tried to vary agitations from 100 rpm to 150, from 150 to 200 and thereby till 250 rpm. So all these were physical requirements that were optimized. Not only that, chemical changes were also optimized. In that, we tried with different carbon sources. Okay, we added 1% glucose in one medium, then we added 1% galactose in another medium, and so on. Sugars like maltose, lactose, fructose, and sucrose were used as carbon source. So once we got the high activity with one particular sugar, further concentration of this sugar were also optimized. So let's say lactose is giving me high production of lipase with respect to as compared to other carbon sources. Then I went with varying the concentration of lactose from 0.1% to 2.5%. So it was an huge experimental where lipase production was optimized. Not only that, further we went with optimizing nitrogen sources. Now in nitrogen sources, I tried with crude nitrogen sources also and synthetic nitrogen sources also, wherein peptone, yeast extract, tryptone, urea, casein and ammonium chloride were used as different nitrogen sources. Again in this also, once we got highest production with a particular nitrogen source, we went in standardizing their concentration. We started with minimum concentration of 0.5% and then slowly and gradually increase the concentration till 2.5%. Not only this, as lipase enzymes they require inducers and generally oils and fatty acids are inducer, we have also used different lipid sources. The lipid sources used in my study include olive oil, palm oil, coconut oil, twin 80 and tributylene. So like we did with carbon and nitrogen source, with nitrogen source we also did the same with lipid source. So once we got a particular lipid source, we tried to differ with the concentration of that source. Let's say coconut oil was giving me a good amount of lipase activity, we went ahead with increasing the concentration from 5% to 2.5%. And when all these parameters were optimized, we mixed this parameter, we brought this parameter together. So we formulated one medium which was having an optimum temperature, optimum pH, optimum agitation, optimum carbon, optimum nitrogen sources and thereby resulting in high amount of enzyme production. All this technology wherein the standardization of enzyme production is done is called as upstream technology. So once we got high amount of lipase enzyme in particularly optimized medium, we went ahead with purifying the lipases. And further, these purified lipases were used in catalyzing esters formation. So the purified lipases were then utilized in catalyzing esterification reaction. Now this reaction has also array of compounds which include use of alcohol and acid. Different combinations of acid and alcohol were used which are giving a distinct note of flavors. I basically tried with acetic acid, formic acid and butyric acid, and further I gradually changed different alcohol. In my study, I had used ethyl alcohol, methyl alcohol, propyl alcohol, and so on. So based on permutation and combination basis, I tried with different concentration and different combination of acid and alcohol to give distinct ester. Now, I kept in mind that although I am changing the combination, the ester must be the particularly of chemical interest. I mean to say that if I want ethyl acetate, I will be using acetic acid and ethyl alcohol. Now, ethyl acetate results in grape or cherry-like flavor. Yes, so whenever we are eating any confectionaries and all, chocolates and all, so the grape-like flavor and cherry-like flavor is because of the artificial essence which are added into that. Now, these essences are made up of chemical and thus they are very harmful. But my research have given a proof of concept and it has given a line that all this chemically synthesized flavoring compound can also be synthesized by biotechnological route by using a biocatalyst, which is a clean and green source of sustainable requirement. So if I go ahead, I have targeted different flavors which include banana flavor. The banana flavor is because of butyl acetate in the essence node, which is giving that typical ripe banana flavor. Now this butyl acetate is formed by adding acetic acid and butyl alcohol in the combination, and the catalysis is done by the enzyme which was lipase in my study. Further, I went to synthesize orange flavor, mushroom flavor, wax flavor, apple flavor, plum flavor, apricot flavor and all these flavors generally I targeted on fruit smell flavors, and they have esters like ethyl acetate, methyl acetate, propyl acetate, isoamyl butyrate and whole lot. In all, 21 combinations one tried. Yes, I tried different combinations and this were around 21 and they were having different categories of esters. And for my surprise, I was very much successful in getting the results also. I was able, out of 21 essays, I was able to get 12 different positive results. And these 12 were different notes of flavor. I was able to synthesize isoamyl acetate, butyl stearate, octyl deconate and all these esters are having application in cosmetic brand, in flavoring, they are used as flavoring agent, they are used in article wherever smell and essence are required. So it was a huge achievement that my research paved a way to have a biotechnologically synthesized flavors. Now further, my idea was also to genetically modify the organism. But although because of the time span in my research journey, I kept it at my future prospectus. I mean to say that this enzyme was synthesized, let's say, in 24 hours. And the flavoring compound was synthesized in next 24 hours. My motto was to reduce the time also by genetic engineering. So we can take the gene from the organism which is able to do the catalysis and synthesize this flavoring compound, and we can genetically modify different microorganisms like E. coli, yeast and all, wherein we can get the same result quicker and in much rapid way. So this is about the future plan of the work. Let me see when I will get a scope to come with this journey coming true too. Lastly, I had used microfluidics. Microfluidics is a blooming science nowadays, although 30 decades the work has been done, but in India, hardly any work has been done in microfluidics. We can, in layman term, I can tell that microfluidics is nothing but lab-on-chip technology, wherein fluids are manipulated at microchannel. Now, being a hardcore researcher being in a lab and all, there was a problem where I noted that there is a huge requirement for reagents, there is a huge requirement for sample in all assay. So even if I have to do a Benedict test, I will require around 1 to 2 ml of sample and 5 ml of Benedict reagent. I will require a lab setup, I will require a test tube, and yes, after putting Benedict's reagent in the sample, we also required to heat them. So we required boiling water bath for the burner requirement is also there. And the result is also some time consuming depending upon how the sample is all and where the Benedict test is been done. It will take around 15 to half an hour to give the result. So there is a huge amount of manpower requirement depending upon the sample. As we can see in a clinical setup in laboratories and all, there is not one sample, there are tremendous sample, and in order to process this tremendous sample, manpower is needed, sophisticated labs are needed, and there involve the use of reagent and sample too. So afterwards it is a wastage, and the waste is also not disposed of easily. It requires autoclaving the waste so that whatever pathogens are there, bacteria are there, that are getting killed, and then the whatever the discard is there, that is safe for environmental disposal. I wanted something which is sustainable, and the solution was given by microfluidics. Microfluidics is another term for lab-on-chip, where all manipulations are done in a single chip which is of very small dimension. The chip can be placed on a fingertip also. And the pioneer of this microfluidics technology is a Whiteside group which is doing a tremendous research in this. And to give a good example of microfluidics platform, I can quote an example of glucometer or the pregnancy strip, which is an on-site diagnosis. So the patient or the layman who has no idea about what the test is about, the person has just to place the sample on the strip, and depending upon the coloration, if the red line is there and two red lines are there, then the test is positive. If one red is there and one is blank, then the test is negative. Such a easy to go platform for rapid and on-site detection is offered by microfluidics. So I tried to include microfluidics in my research, and I also tried to reduce the use of substrate. Generally, microfluidics technology requires PDMS, it requires silicon wafers and a huge amount of substrate, which is adding cost of the device. I tried to use an eco-friendly sustainable solution which was offered by a paper. Yes, you are right, it is a paper, and in lab and in general science there are many papers. For example, Whatman filter paper, cellulose paper, nitrocellulose paper, nitrocellulose acetate paper and all. Now paper is lightweight, it is easily available, it is inexpensive. A whole sheet of paper is hardly costing some bucks, and we have to use hardly 1 to 1 centimeter of that paper. So even if I have one sheet, I can make around 100 to 200 devices from a single Whatman paper sheet. Plus requirement is low. So wherever Benedict reagent required sample of around, let's say 5 ml, my assays are using sample hardly nanoliters. Okay, I did my research in nano and microliter by utilizing around 5 microliter of samples and reagents. So sample requirement is also less. So you have seen that glucometer. Glucometer does the diagnosis of sugar by hardly 1 drop of blood. So we can see that it is it is on-site, it is taking very less amount of sample, and the results are readable on-site, so that lay person, those who have no idea about the thing also can monitor the result. Also the energy consumption is less. If I do the same thing in laboratory, I will require all facilities like incubator, autoclave, burner. So energy consumption is more in laboratory. However, when we use microfluidics platform, it requires hardly energy, and I had used paper as the substrate, which required least energy. And once the device has been made, I can transport this paper made devices into remote location also, where the poor people cannot afford hi-fi laboratory facilities. And being a paper, it is safe for disposable also. We can just flame the paper and dispose it off, so that it is not leaving any hazardous substance in the environment. So with this motto, I had used paper-based substrate to develop my microfluidics devices. And thus the paper is giving a assured detection device. Now this assured word I can elaborate as affordable. That means the paper-based microfluidics devices are affordable. Sensitivity is like that which is done in clinical laboratory. We can bring the sensitivity by incorporating antibodies and all. Since antibodies will be used, the devices are specific also. They are user friendly, so that any person who is having least knowledge can also make use of this device, and the results are rapid and robust. We need to wait for 24 hours for result to come. We can detect the result on site and at site. And yes, they are equipment friendly. They doesn't require any equipment like instruments like microscope or autoclave or hot air oven or incubator, and the chip can be delivered to the end users. So by utilizing microfluidics, I was able to detect lipase activity on site. So generally detection of lipase activity, as I said, I had used colorimetric and titrimetric method, and all involved use of huge amount of reagent and sample. Also there was a need to use lipase in a huge amount. But since these lipases are they are synthesized in very minute amount, there was a need for this microfluidics to inculcate in this project. So in a bid to enhance the efficiency and precision, microfluidics technology was incorporated for rapid on-site detection of lipase. This cutting-edge technology allowed for real-time analysis and enabling swift identification and characterization of flavor compound. Further, I tried to incorporate smartphone technology in this. Yes, so the person did not go to doctor also. Just by clicking the photographs of the result on the chip at the home, this photos can be made available at the doctor's house by smartphone application, and the doctor can give the finding. So integrated with the smartphone application, the microfluidics device provided an accurate concentration analysis and further enhancing the utility. Yeah. Looking ahead, our research paved a way of future prospectus. As I have said, that optimizing production using solid state fermentation was also a need, but because of time constraint, I have not done that. But I always will try to do if I got the opportunity to work on the same line. And further, I will employ genetic manipulation for strain improvement. The extraction and purification of esters also hold a tremendous potential. So whatever esters were produced, they were in a crude form. Okay. In future studies, I would like to purify this ester so that they will be at the peak note of the flavor. And ultimately the made chip, I would like to commercialize it. Now I am thrilled to share the outcomes of my research journey, which are documented in various esteemed journals. My first publication was on, it is a review article on paper microfluidics in microbial world. Okay, it has around 1 citation and it has reached around 161 reads and all. But this publication, which is a review article, I have tried to inculcate all technologies which was utilized in making chip. My next publication is on synthesis of flavoring compound using microbial lipases. So whatever 8 to 9 esters was formed by me, I had testamented in Journal of Advanced Scientific Research. The paper is easily available. It is freely costing. It doesn't involve any purchase cost and all. So you can surely read this paper, wherein you can get a deep idea about my protocols, what formulation, what were the concentration, and how these flavors were extracted and further noted. The optimization of lipase activity from Staphylococcus gallerium, the one of the microorganism which I was able to sequence, and its partial purification is also available in the journal, and all these are also available in the ResearchGate portal. Now I am deeply thankful to my mentor, Dr. Meenal Dukhande Madam from Department of Microbiology of Guru Nanak Khalsa College, whose guidance, wisdom and unwearying support have been the guiding light for this research expedition. Your expertise and mentorship have not only enriched my academic journey, but have also inspired me to push the boundary of the scientific inquiry. Thank you.
ज्ञानेन सदृशं पवित्रमिह विद्यते नहि ज्ञानेन सदृशं पवित्रमिह विद्यते नहि ज्ञानेन सदृशं पवित्रमिह विद्यते